Hello everyone, my name is Sarah Ash and today I will be presenting my research on the reaction of S. aureus to soils cultivated around Rochester, New York. This is the organization of my presentation, and in my introduction I will be covering background, rationale, and objective. Our interaction with bacteria is inevitable and will always be a part of our everyday life. Bacteria are found everywhere in the world, even in our own body. Many bacteria in the human body form a mutualistic relationship with the body by helping digest food and producing essential vitamins. Occasionally, though, foreign bacteria will enter the body and cause sickness. The GI tract is an essential part of the immune system that helps to defend the body against these intrusive bacteria. It does so by producing hydrochloric acid, which creates an acidic environment which is known to suppress bacteria. Sometimes, though, the GI tract will not be sufficient enough with this defense against bacteria, for one reason or another. This is when antibiotics come into play. Antibiotics typically come from natural products that are made by bacteria and are considered either bactericidal, meaning that it kills bacteria, or bacteriostatic, meaning that it only stops the cell's growth. These products are usually made by bacteria with the intent to kill or deter other microorganisms in environments with competition for food or other resources. Once these natural products have been isolated, they are usually grown in low-throughput fermentation conditions. Bacteria can naturally gain and lose traits of resistance. This, paired with the misuse of antibiotics by not using them for the proper amount of time, puts selective pressure on the bacteria, causing those with gene of resistance to thrive. These bacteria can also share their gene for resistance through horizontal gene transfer. Antibiotic resistance is becoming a large problem, causing an increasing amount of fatalities. Many bacteria have consistently increasing rates of resistance. Luckily, scientists are looking towards innovative methods of research. For example, elaticins are a new antibiotic candidate and are discovered via cell biome screening, rather than the typical cell culturing. This is especially useful because most bacteria are not culturable. The CDC has deemed many bacteria as serious or urgent threats, including C. diff and Staphylococcus aureus. Resistance rates are the amount of bacteria that possess a gene that resists antibiotic treatment. The rate of resistance against the antibiotics tetracycline and ciprofloxacin have increased from around 17 and 9% in 2009 to around 24 and 25 and 23% in 2015, respectively. I chose to analyze soil bacteria primarily because the majority of antibiotics in use are derived from soil bacteria. I was also researching antibiotic properties of soil and read a paper that mentioned that people in Jordan, both past and present, use red soil to cure skin infections like diaper rash. The study showed that the soil was able to cause a decrease in both M. luteus and S. aureus. The first step in my methods was to measure off a 33 cm by 33 cm quadrat. I then took 4 corner samples using a soil core and took the top 3 centimeters of each sample. I did this at 5 locations, and at every sampling location I recorded data such as soil type, temperature, latitude, and longitude, and the time of collection. After I collected all of my samples, I individually sieved them, and I used a 4 millimeter and a 1 milliliter sieve. Then I made nutrient broth by mixing 300 milliliters of distilled water and 3.9 grams of powdered nutrient agar. I then put 10 milliliters of the mixture into 20 tubes. Then I mixed 1 gram of each sample with 100 grams of distilled water and added 10 milliliters of each sample to a tube with broth. Then I took aliquots every 2 days over a 6-day span, and I froze the aliquots after taking them. After I collected all of my aliquots, I sent them to an external lab for analysis and waited for the results. This is the flowchart that I used to determine soil type. It's from the USDA, and it's titled Guide to Texture by Feel. This is an example of how I organized my sample names and picked which samples to keep at 20 degrees Celsius and which samples to keep at 37 degrees Celsius. It also lists what day I took the aliquots and what I named the aliquots. This map shows the 5 locations that I took samples. Number 1 being Durand Beach, 2 being Arundequoit Cemetery, 3 being Loftus Playground, 4 being Highland Park, and 5 being Genesee River. The table at the bottom displays the information that I took after sampling at each location. The results of this experiment are pretty cut and dry. The positive control, which used an extract from the lab it was analyzed in called G192, showed a zone of inhibition. None of the other 120 screening results, on the other hand, did. There are a plethora of possible reasons that results from the aliquot analysis show no antimicrobial properties. S. aureus is a gram-positive bacteria, and it is possible that the aliquots tested produce natural products that target only gram-negative bacteria. It is also possible that the aliquots secreted natural products that affect a narrow spectrum of bacteria, and S. aureus is not one of them. A variety of issues could have also stemmed from the methodology used, and it may have resulted in the experiment not being efficient enough to allow the bacteria present to secrete enough or any antimicrobial natural products at all. The bacteria in the samples also may have been able to sense a difference in their environment, and their response may have been to cease the production of natural products. Lastly, the time from the collection of the soil samples to the time of the analysis of the aliquots may have been too long for the bacteria to survive due to temperature differences or other external factors such as light exposure or humidity. In the future, it may be feasible to shorten the time between sample collection and analysis. It also may be beneficial to test aliquots against multiple types of bacteria instead of just one. Thank you.